Hypoxia rewires membrane lipids, drives pancreatic cells to move. Pancreatic cancers are aggressively and deadly with high rates of metastasis and poor prognosis. The tumor environment is also hypoxic. The cells rapidly divide and thrive in very low oxygen condition. Now IIT Bombay researchers have shown there the hypoxia ends up enhancing the cells metastatic behavior by affecting the cells plasma membrane lipids. Hypoxic conditions can help the cells migrate more. The membrane is not just something that is covering the cell and keeping everything that should be inside and everything that should be outside, said Beats Pilani. Biophysicist Sudipta Maiti, who was not involved in the work, it is also the cell's window to the outside world. Stiff yet malleable. In two studies published in 2023 and 2025, IIT Bombay chemist Sovana Kapoor and her colleagues showed that hypoxia can modify the kinds of lipids present in the cancer cells plasma membrane, thus affecting how easily the cells are able to fluidize and move around. In the 2023 Biochemia at Biophysics Act paper, they reported that hypoxia causes the cells of the PANC1 pancreatic cancer cells line to migrate more. This happened because hypoxia was able to modulate the lipid ratios in the cells. Lipids that promote membrane stiffening were pushed towards the cytoplasm and inner organelles. They also observed that the cells had less cortical stiffness, meaning the layer of skeleton just below the membrane allowed for more fluidity. In the Journal of Membrane Biology study published in August, the researchers studied a different cell line called CAPAN2. In contrast, researchers found that this pancreatic cancer cell line had higher cortical stiffness in response to hypoxia, but it still maintained its malleable nature by adding more membrane components to its plasma membrane. Even the lipidome modifications were different in this case, with some lipids that promote membrane stiffening making their way to the plasma membrane, but the scientists believe that this is a local stiffening effect and doesn't affect the overall membrane properties. Even though hypoxia affected cell stiffness differently in the two pancreatic cancer cell lines they studied, the cells still modified their own membrane in a way that their migration increased when oxygen was low. Together, the studies could open new pathways that could both contribute to and prevent excessive cell migration in pancreatic cancer cells, and modulating them could potentially help mitigate the tumor from metastasizing. When Dr. Kapoor and her student initially realized when Dr. Kapoor and her student initially realized that Hypoxia caused the PANC1 cells to migrate faster. They decided to check how the membrane biophysical properties, like how ordered, fluid or bent the membrane was, were changing. But they were for a surprise when they found that some board properties of the membrane did not change dramatically. Then we decided, let us then not look at ensemble properties of the membrane. Maybe let us look at the composition of the membrane, Dr. Kapoor said, and then we realized that actually the hypoxia is changing the lipidome of the cell. They found that even though the cell seemed to be increasing its amounts of membrane stiffening lipids, like lipids with long fatty acid chains or Saturday bonds, the lipids in the plasma membrane did not change as much. There is a feedback loop that is going on which helps the membrane to maintain homeostasis so that the membrane properties remain the same, said Dr. Kapoor. The changes that the hypoxia brings about in the lipid levels, they get compensated in internal organelles like the nucleus, mitochondria, and endoplasmic reticulum. They also found that the PANC1 cells had less cortical stiffness because of how action volume with low stiffness and any membrane stiffening lipids being trafficked inwards, the cell could maintain a malleable membrane that helped it migrate, the researchers speculated. The story is slightly different for CAPN2. Hypoxia still helped the cell migrate more, but their cortical stiffness was higher in hypoxic conditions. But other experiments revealed that the cell was trafficking more membrane material to the plasma membrane to counteract this stiffness and likely maintain its migratory behavior. The researchers also found an increase in saturated lipids in the plasma membrane, suggesting that the cell may also be trying to locally enhance the membrane stiffness. The seems to have some diversity from one cell type to another, said Mohammed Salim, a biophysicist at the National Institute of Science, Education and Research, Bhubaneswar. Dr. Maiti said solid state nuclear magnetic resonance, a technique that use magnetic fields to look at the atomic level structure and dynamics of solid molecules, could probe deeper into the plasma membrane and figure out how exactly hypoxia could be changing its properties. Lipids molecules are like tadpoles. It is a little head and a little tail, and the whole thing is probably 2 nanometers long. Solid state NMR can look at this little tail and see how dynamic it is be explained. It can be fluctuating madly or pretty stable because the lipids are packed. Translate into mechanical property, how stiff something, how loose something. It will be nice to show quantitatively using solid state MMR how hypoxia leads to change in this membrane order. Effects on other cancers. Both Dr. Kapoor and Dr. Salem said that going forward, one must also explore how hypoxia affect other cancers. Each of these cancers cell type has their own niche and microenvironments. Dr. Salim said it would be interesting how hypoxia and the difference in those microenvironments could come into play to drive their migration. A small biochemical reaction can include a larger physical manipulation of the cell membrane. This could also open up venues for exploring membrane targeted anti cancer